Morphogen gradients. One of the most important mechanisms governing cell fate specification involves gradients of paracrine factors that regulate gene expression. Such signaling molecules are called morphogens. A morphogen is a diffusible biochemical molecule that can determine the fate of a cell by its concentration. That is, cells exposed to high levels of morphogen activate different genes than those cells exposed to lower levels. Morphogens can be transcriptional factors produced within a syncytium of nuclei, as in the Drosophila blastoderm. They can also be paracrine factors that are produced in one group of cells and then travel to another population of cells, specifying the target cells to have similar or different fates according to the concentration of the morphogen. So, uncommitted cells exposed to high concentrations of the morphogen nearest its source of production are specified as one cell type. When the morphogen's concentration drops below a certain threshold, a different cell fate is specified. When the concentration falls even lower, a cell that initially was of the same uncommitted type is specified in yet a third distinct manner. So, regulation by gradients of paracrine factor concentration was elegantly demonstrated by the specification of different mesodermal cell types in the frog Xenopus levis by activin, a paracrine factor of the TGF-beta superfamily. Activin secreted beads were placed on unspecialized cells, specified cells from the early Xenopus embryo. The activin then diffused from the beads. At high concentration, activin induced expression of guscoid gene, whose product is a transcription factor that specifies the frog's dorsalmost structures. At slightly lower concentrations of activin, the same tissue activated the XBRA gene and was specified to become muscle. At still lower concentrations, these genes were not activated, and the default gene expression instructed the cells to become blood vessels and heart. So, the range of paracrine factor and thus the shape of its morphogen gradient depends on several aspects of that factor synthesis, transport, and degradation. In some cases, cell surface molecules stabilize the paracrine factor and aid in its diffusion, while in other cases, cell surface moieties retard diffusion and enhance degradation. Such diffusion regulating interactions between morphogens and extracellular matrix factors are very important in continuing or coordinating organ growth and shape. Paracrine families. Next major topic is gametogenesis in animals. One of the most important events in sex determination is gametogenesis, the differentiation of germ cells into gametes, eggs, and sperm. And it is the primordial germ cells that are the bipotential precursors of both eggs and sperm. If they reside in the ovaries, they become eggs, and if they reside in the testes, they become sperm. Primordial germ cells. All these decisions are coordinated by factors produced by the developing gonads. One of the most amazing things about germ cells is that they provide the continuity between generations. The adult body perishes, but the germline forms the gametes that create a new body which will also perish. The immature sperm or eggs in your body has come from a germ cell lineage that has resided in the gonads of reptiles, amphibians, fish, and invertebrates. Second, and importantly, the cells that generate the sperm or eggs do not originally form inside the gonads. In Drosophila and mammals, they form in the posterior portion of the embryo and migrate into the gonad. This pattern is common throughout the animal kingdom. The germ cells are set aside from the rest of the embryo, and the cell's transcription and translation are shut down while they migrate from peripheral sites in the embryo to the gonad. It is as if the germ cell were a separate entity reserved for the next generation, and repressing gene expression makes them insensitive to the intracellular commerce going on all around them. So, although the mechanisms used to specify the germ cells vary enormously across the animal kingdom, the proteins expressed by germ cells to suppress gene expression are remarkably conserved. These proteins, which include the VASA, NANOS, TUDOR, and PV family proteins, VASA, NANOS, TUDUR, then PIWI family proteins, can be seen in the germ cells of cnidarians, fleas, and mammals. VASA proteins, which appear to activate germ cell specific genes, are required for germ cells in nearly all animal studies. Nanos is involved in repressing translation and preventing cell death. Equally remarkable is that the signal that induces the formation of PGCs also appears to be conserved throughout the animal kingdom. In mammals and those insects that induce germ cells, BMP signals are required for the formation of PGCs. In mammals, BMP4 from the extra embryonic cell layers induce mesenchymal cells to produce PGCs by activating those genes that specify the cells to be germline, while simultaneously blocking the expression of those genes that prevent cells from becoming part of the germline. And then this strategy of simultaneously activating one set of genes while repressing others is similar to that seen in mammalian gonadal sex determination. Migration of PGCs to endoderm. Amnion neural fold allantois PGCs embryo. Migration of PGCs into gonad, genital ridges, hindgut, somite, primordial germ cells, dorsal mesentery, cloaca, mesonephros, then genital ridges. Then PGCs in mammals from genital ridge to gonads. In mammals, the newly formed PGCs first enter into the hindgut and eventually migrate forward and into the bipotential gonads, multiplying as they migrate from the time of their specification until they enter the genital ridges. The PGC are surrounded by cells secreting stem cell factor or SCF. So SCF is necessary for PGS motility and survival. Moreover, the cluster of SCF secreting cells appear to migrate with the PGCs, forming a traveling niche of cells that support the persistence, division, and movement of the PGCs. Once they are in the gonad, these cells are sustained by BMPs that create a niche for them in the genital ridge. The PGCs are then told by the gonad whether to initiate oogenesis, the formation of eggs, or to initiate spermatogenesis, the formation of sperm. A fundamental difference between mammalian males and females involves the timing of meiosis. In females, meiosis begins in the embryonic gonads. In males, meiosis is not initiated until puberty. The gatekeeper for meiosis appears to be the STRA8 transcription factor, which promote a new round of DNA synthesis and meiotic initiation in the germ cells. In the developing ovaries, STRA8 is upregulated by two factors, WNT4 and the retinoic acid RA coming from the adjacent kidney. So, in the developing testis, however, STRA8 is downregulated by FGF9 and the retinoic acid produced by the developing kidney is degraded by the testis secretion of the RA degrading enzyme CYP26B1. During male puberty, moreover, retinoic acid is synthesized in the Sertoli cells and induces STRA8 in stem cells. Once STRA8 is present, the sperm progenitor cells become committed to meiosis. Meiosis, the intervening of life cycles. Meiosis is perhaps the most evolutionary invention of eukaryotes, for it is the mechanism for the transmission of genes from one generation to the next and for the recombination of sperm and egg-derived genes into new combinations of alleles. So, one Benedin's 1883 observation that the division of germ cells caused the resulting gametes to contain half the diploid number of chromosomes demonstrated that the chromosomes of the offspring are derived in equal numbers from the nuclei of the two conjugated half germ cells and hence equally from two parents. So meiosis is a critical starting and ending point in the cycle of life. Sexual reproduction, evolutionary variation, and the transmission of traits from one generation to the next all come down to meiosis. So to understand what germ cells do, we must first understand meiosis. Meiosis is the means by which the gametes halve the number of their chromosomes, just turned into half. In the haploid condition, each cell has only one copy of each chromosome, whereas diploid cells have two copies of each chromosome. This feat is accomplished by the occurrence of a single round of DNA multiplication followed by two successive chromosomal divisions. After the germ cell's final meiotic division, the period of DNA synthesis occurs so that the cell initiating meiosis doubles the amount of DNA in its nucleus. In this state, each chromosome consists of two sister chromatids attached at a common kinetochore. In the first of the two meiotic divisions, homologous chromosomes, for example, the two copies of chromosome 3 in the diploid cell, come together and are then separated into different cells. Since the first meiotic division splits homologous chromosomes between two daughter cells such that each daughter cell has only one copy of each chromosome. These cells are therefore haploid. But each of these chromosomes has already replicated. So the second division separates the two sister chromatids from each other. The net result of meiosis is 4 cells, each with a haploid set of unreplicated chromosomes. During the next stage of meiotic prophase, pachytene, the chromatids thicker and shorter, individual chromatids cannot be distinguished under the light microscope and crossing over may occur. The next is spermatogenesis in mammals. Spermatogenesis, the developmental pathway from germ cell to mature sperm, begins at puberty and occurs in the recesses between the Sertoli cells. Spermatogenesis is divided into three major phases. A proliferative phase. Where sperm stem cells, spermatogonia, increase by mitosis. So a proliferative phase where sperm stem cells, spermatogonia, increase by mitosis. A meiotic phase involving the two divisions that create the haploid state. A meiotic phase involving the two divisions that create the haploid state. A post-meiotic shaping phase called spermiogenesis, during which the round cells, spermatids, eject most of their cytoplasm and become streamlined sperm. As different epididymis, testis, speminiferous tubules. Sertoli cells are there. Type B spermatogonium is there. Type A spermatogonium. Primary spermatocyte, secondary spermatocyte, spermatid. Then lumen of tubule, sperm residual body. So this is a picture which depicts sperm maturation, cross section of the seminiferous tubule. Spermatogonia are blue. Spermatocysts are lavender. And the mature sperm appear yellow. Then simplified diagram of portion of a seminiferous tubule illustrating relationship between spermatogonia, spermatocyte and sperm. As these germ cells mature, they progress towards the lumen of the seminiferous tubule. The proliferative phase begins when the mammalian PGCs arrive at the genital ridge of the male embryo. Here they are called gonocytes and become incorporated into the sex coats that will become the seminiferous tubules. The gonocytes become undifferentiated spermatogonia residing near the basal edge of the tubular cell. These are true stem cells in that they can re-establish spermatogenesis when transferred into mice whose sperm production has been eliminated by toxic chemicals. Spermatogonia appear to take up residence in stem cell niches at the junction of the Sertoli cells, the epithelium of the seminiferous tubule, the interstitial testosterone producing Leydig cells, and the testicular blood vessels. Adhesion molecules join the spermatogonia directly to the Sertoli cells, which will nourish the developing sperm. The percentage of the conocytes that become true stem cells probably differs greatly among groups of mammals, and the cell defining the cell stem cell niche may also differ. This is because different groups of mammals have different strategies for sperm production. Mice have 12 amplifying divisions of progenitor cells between the sperm stem cell, and spermatocyte that undergoes meiosis. They produce 40 million sperms per gram of testis tissues per day. Humans produce more stem cells but have only 5 transit amplifying divisions between the stem cell and the spermatocyte. Men generate 4.4 million sperm per gram of testis tissue each day. Although this is a tenfold less efficient than mice, it means that adult human males generate more than 1,000 sperm per second. Each day, some 100 million sperms are made in each human testicle, and each ejaculate releases 200 million sperm. Unused sperm are either reabsorbed or passed out of the body in urine. So during his lifetime, a human male can produce 10 raised to 12 to 10 raised to 13 sperm. The meiotic proliferation of the stem cells produce type A spermatogonia, which are held together by the cytoplasmic bridges. However, these bridges are fragile, and a cell can split off from the group and become a stem cell again. Then, glial derived neurotrophic factor, or GDNF, secreted from the Sertoli cells, keeps the stem cells in mitosis. However, the BMPs and WNTs start inducing the type A spermatogonia to differentiate further to sperm. Then the meiotic phase, getting to haploid spermatids. Type B spermatogonia are the precursors of spermatocytes and contain high levels of STRA8. These are last cells of that lineage to undergo mitosis, and they divide once to generate the primary spermatocytes, the cells that enter meiosis. Each primary spermatocyte undergoes the first meiotic division to yield a pair of haploid secondary spermatocytes, which complete the second division of meiosis. The cells thus formed are called spermatids, and they are still connected to one another through their cytoplasmic bridges. The spermatids that are connected in this manner have haploid nuclei but are functionally diploid, since a gene product made in one cell can readily diffuse into the cytoplasm of its neighbors. During the divisions from undifferentiated spermatogonia to spermatids, the cells move further and further away from the basal lamina of the seminiferous tubule and closer to its lumen. Then the post-meiotic phase, spermiogenesis. As the spermatids move towards the lumen, they lose their cytoplasmic connections and differentiate into spermatozoa, a process called spermiogenesis. In humans, the progression from spermatogonial stem cells to mature spermatozoa takes 65 days, and the last third of it, about 21 days, is taken by the spermiogenesis. The mammalian haploid spermatid is a round, unflagellated cell that looks nothing like the mature vertebrate sperm. For fertilization to occur, the sperm has to meet and bind with an egg, and the spermiogenesis prepares the stem for these actions of motility and interaction. The process of mammalian sperm differentiation is important. Then next, oogenesis. Scientists who study oogenesis often write in terms of the wonder that the process generates and the huge unknown questions that remain to be solved. Mammalian oogenesis, or egg production, differs greatly from spermatogenesis. The eggs mature through a symphonic coordination of hormones, paracrine factors, enzymes, chromatin structures, and tissue anatomy. So, mammalian egg maturation can be seen as having 4 stages. First, there is a stage of proliferation. In the human embryo, the 1,000 or so PGC reaching the developing ovary divide rapidly from the 2nd to the 7th month of gestation. They generate roughly 7 million oogonia. While most of this oogonia die soon afterwards, the surviving population, under the influence of retinoic acid, enter the next step and initiate the first meiotic division. They become primarily oocytes. This first meiotic division does not proceed very far, and the primary oocyte remain in the diploidine stage of first meiotic prophase. This prolonged diploidine stage is sometimes referred to as the dictate resting stage, and it may last from 12 to 40 days. With the onset of puberty, groups of oocytes periodically resume meiosis. At that time, the luteinizing hormone from the pituitary gland releases this block and permits these oocytes to resume meiotic division. They complete first meiotic division, and the resulting secondary oocytes proceed to second meiotic metaphase and undergo maturation steps. This maturation involves the crosstalk of paracrine factors between the oocyte and its follicle cells, both of which are maturing during this phase. The follicle cells activate the translation of stored oocyte mRNA encoding proteins such as the sperm binding proteins that will be used for fertilization and the cyclins that control embryonic cell division. After the secondary oocyte is released from the ovary, meiosis will resume only if fertilization occurs. At fertilization, calcium ions are released in the egg, and these calcium ions release the inhibitory block and allow the haploid nucleus to form. Then oogenic meiosis. An oocyte is a remarkable cell with two universal roles in reproduction: correct segregation of chromosomes during two successive rounds of meiosis and sustaining viability of early embryo until transcription activation. To endow the early embryo with the ability to meet its developmental demands before the nuclear genome is activated in the embryonic cells, oocytes grow to a large size and undergo divisions that minimize the loss of cytoplasm. While the sperm loses its cytoplasm, the egg accumulates cytoplasm. Then oogenic meiosis in mammals differ from spermatogenic meiosis in numerous ways. An oogenic meiosis in mammals differs from spermatogenic meiosis in numerous ways. First, when the primary oocyte divides, its nuclear envelope breaks down and the metaphase spindle migrates to the cortex or periphery of the cell. At the cortex, an oocyte-specific tubulin mediates the separation of chromosomes, and mutations in this tubulin have been found to cause infertility. At telophase, while both the daughter cells now contain a haploid nucleus, one contains very little cytoplasm, while the other remains nearly the entire volume of cellular constituents. The smaller cell becomes the first polar body, and the larger cell is referred to as the secondary oocyte. This asymmetrical cytokinesis is directed through a cytoskeleton network composed chiefly of filamentous actin that cradles the mitotic spindle and brings it to the oocyte cortex by myosin mediated contraction. A similar unequal cytokinesis takes place during the second division of meiosis. Most of the cytoplasm is retained by the mature egg or the ova. The second polar body forms but receives a little more than a haploid nucleus. In humans, the first polar body usually does not divide. It undergoes apoptosis around 20 hours after the first meiotic division. This oogenic meiosis conserves the volume of oocyte cytoplasm in the single cell rather than splitting it equally among four progeny. A second way in which oogenic meiosis in mammals differ from spermatogenetic meiosis is in the mechanics of meiosis 1. The oocyte meiotic spindle lacks centrioles. Rather, the microtubules of the meiotic spindle are organized by mRNAs and enzymes located on the chromosome and the spindle fiber themselves. Instead of two centrioles, numerous microtubule organizing centers, MTOCs, form around the nuclear envelope at first meiotic prophase. The MTOs coalesce at the future spindle poles once the nuclear envelope breaks down. Oocytes and age. The retention of the oocyte in the ovary for decades has profound medical implications. Most human embryos do not survive to birth. A large proportion, perhaps even a majority of fertilized human eggs have too few chromosomes to survive. Genetic analysis has shown that such aneuploid, incorrect number of chromosomes, is usually due to errors in oocyte meiosis. So only a few aneuploids, such as those of the sex chromosomes and chromosome 21, survive to be born, and the percentage of babies born with such aneuploidies increase greatly with maternal age. Women in their 20s have only a 2 to 3% chance of bearing a fetus whose cells contain an extra chromosome. This risk goes to 35% of women who become pregnant in their 40s, a great amount of difference. The reason for this appears is due to the gradual loss of cohesin proteins from the chromosomes as the cell ages, causing a less stable linkage between the kinetochore and spindle during meiosis metaphase. Why is that happening? So, gradual loss of cohesin protein from the chromosome as the cell ages, causing a less stable linkage between the kinetochore and spindle during mitosis metaphase.